Assalamu alaikum. Dr. Mariam Ashraf, Anatomy Department. And today's lecture is in continuation to the previous lectures on the development of the central nervous system. And we left off in the last lecture on how the telencephalon or the forebrain develops. Now, before I explain this diagram, I just want you to follow this blue line here like this. This area, this entire area is the diencephalon, the thalamus, the hypothalamic sulcus, pineal thickening, and other structures like epithalamus. Now, this area I told you is embedded in the medial and inferior aspect of each hemisphere. And this previously discussed in the development of the diencephalon, we said the roof plate of the diencephalon, anteriorly it forms the choroid plexus of the third ventricle, which is the ventricle of the diencephalon. And posteriorly, there is a thickening and you have the pineal body here. Now, this area that you see is actually lateral to the structures of the developing diencephalon. So, each hemisphere develops lateral to the structures of the diencephalon, and this is made possible by waves of neuroblasts which are extending from the lateral aspect of the diencephalon outwards like this. This is not a 3D diagram, so you have to use your imagination a little bit. And what they've done is that in the diagram B, they've actually shown you a cross section, which is this dotted line here. And if you follow this, this is actually going to correlate with the roof of the diencephalon. And this is the choroid plexus. And these structures that you see here are all going to grow outwardly and away from the ventricle, which is the third ventricle, which is the ventricle of the diencephalon. And so this area and this area, movement away from it will be the developing cerebral hemispheres. Don't look at these details, we will talk about them later on. So the cerebral hemispheres will grow outwards and laterally to the diencephalon. Now I just want to go to this part. The telencephalon, we know that it consists of two portions, the cerebral hemispheres, which are two, one on each side, and a structure called lamina terminalis. Diencephalon we've already discussed. Now we're talking about that part of the telencephalon, which is cerebral hemispheres. When you talk about the development of the cerebral hemispheres, there is a midline structure called lamina terminalis, and it is a thickening between the developing hemispheres. I think they've shown this structure here. This is lamina terminalis, and this is a thickening in the midline. And it is important because a lot of fibers or commissures or bundles of white matter cross through this lamina terminalis from one hemisphere to the other, which we will discuss a little bit later on, but for now you need to understand that when you talk about the development of the cerebral hemispheres, you have to talk about neuroblasts going laterally and outwards from the diencephalon, and then you have to talk about a midline structure which is one on either side of the hemispheres. So each hemisphere will have one lamina terminalis, which is a midline thickening. The junction of the cerebral hemisphere with the roof of the diencephalon does not have neuroblasts. Only ependymal cells and vascular mesenchyme, which will form the choroid plexus of each ventricle. And which ventricle are we talking about? We are talking about the lateral ventricle. The third ventricle is the ventricle of the diencephalon, as you can see here. This is the ventricle of the diencephalon, the third ventricle. Each hemisphere, within each hemisphere, you will have one ventricle, which is called lateral ventricle. And I told you that the cerebral hemispheres will develop lateral to the diencephalon. So at the junction of the roof of the diencephalon and cerebral hemispheres, you will not have neuroblasts, you will only have ependymal cells and vascular mesenchyme, because here you will have formation of the choroid plexus of each lateral ventricle. It will become a little clearer to you as we go on. This diagram, I want to show you exactly this area was the developing cerebral hemisphere. This is the diencephalon. This is the lateral ventricle, one in each hemisphere. Okay. This is the area of the diencephalon. They've labeled one structure, which is thalamus. And this is a green area which is part of the cerebral hemisphere, but we call this corpus striatum. So this is the developing hemisphere, this is the diencephalon, this is the cavity of the hemisphere, which is lateral ventricle, and this is also part of the cerebral hemisphere, but it is colored in green and labeled as corpus striatum. This part, the green area of the cerebral hemisphere, it grows into the lumen of the third ventricle, which means the lumen of the diencephalon. And this has a striated appearance, and so this part of the cerebral hemisphere is called corpus striatum. Now I'll just go back and show you corpus striatum. This is the part of the cerebral hemispheres which are bulging into the cavity of the third ventricle, and this thickening or development of cerebral hemispheres is known as corpus striatum. And it is called corpus striatum because it has a striated appearance. So now we know that the cerebral hemispheres grow outwards and laterally from the diencephalon. The cavity of each hemisphere is a lateral ventricle. Bulging into the cavity of the diencephalon, which is the third ventricle, is a part of the cortex which has a striated appearance, and it is known as corpus striatum. And we also know that there is something called lamina terminalis, which is a midline thickening. Now this is again a section of the hemispheres, and what they've shown you here is the lateral ventricle of each side. So they've shown both hemispheres. It's a cross section. This is the thalamus. This is the caudate nucleus. This is the lentiform nucleus. This is the third ventricle. You can see on either side you have structures of diencephalon. This is third ventricle. Now I just want you to pay attention to the yellow structure and to the purple or reddish purple structure. This striated area is the corpus striatum. This corpus striatum is the part of the cerebral hemisphere which bulges into the cavity of the third ventricle, and it also divides this yellow area from this purple reddish area. And together these structures are known as basal ganglia. Basal ganglia are very important nuclei embedded within each cerebral hemisphere, and they have to do with motor activity. So the corpus striatum divides into the basal ganglia. The corpus striatum divides the basal ganglia into a dorsomedial portion, which is called the caudate nucleus, and a ventrolateral portion, which is the lentiform nucleus, and it is divided into putamen. And we don't need to do these details right now. We will discuss this in neuroanatomy. For now, you need to understand that the basal ganglia are a collection of nuclei in each cerebral hemisphere which are connected with motor activity, and they are divided by corpus striatum here. Now you can see some structure labeling here. You can see neopallium and paleopallium. There are three phylogenetically. There are three types of cortex that we talk about. Paleopallium or paleocortex is the oldest part. Archipallium or archicortex is the area around the olfactory cortex. And the neopallium or the neocortex is the newest form of cortex, and 90% of the structure of the cortex is derived from neopallium. So this is just developmental or anthropological information. Paleopallium is the oldest part of the cortex. Archipallium or archicortex is the area of the olfactory cortex. And neopallium is what most of the cortex and the structure of the hemispheres is made up of. There is another term that you are familiar with, which is called placode. Placodes, ectodermal placodes. Placodes are a combination of ectodermal thickening and neural crest cells, and they have to do with sensory organs which are developing in the head and neck region. Like the olfactory placode, it has to do with the development of the nose, the lens placode for the eye, and the otic placode for development of the ear. So placodes are actually ectodermal thickenings with neural crest cell migration, and they are thickenings concerned with development of sensory apparatus. Now all this gray matter that you have paleopallium, you have archipallium, you have neopallium, then you have a type of cortex which is called corpus striatum. All of this is gray matter. The white matter of the cerebral hemispheres is described as bundles which can travel within the same hemisphere, or they can travel from one hemisphere to the other hemisphere, or they can connect different regions to one another. So commissure is a term which is used to describe the bundles of white matter in each cerebral hemisphere. And in this regard, the lamina terminalis has an importance because the lamina terminalis uh, is sort of like it's a motorway for these commissures which are going to pass from one hemisphere to the other. Now, the detail of the hemispheres we will do when we go to the development of, when we discuss the subject of neuroanatomy. But for now, it is enough for you to know that white matter or bundles of fibers connecting different parts of the same hemisphere or connecting one part of the hemisphere with the other, they are called commissures. Now, this is a picture where I want to show you some of the commissures that develop. You don't need to do the detail right now, but this is the posterior commissure, this is the habenular commissure, the corpus callosum, this dotted line that you see here. This corpus callosum is the most important commissure to develop, and it connects one hemisphere to the other. Then you have the anterior commissure. The optic chiasma is also fiber bundles, so that's a commissure. So the white matter, whenever it gets together as a bundle in the cerebral hemispheres, it is known as commissures. And please notice there's a small mistake in the heading because commissures has a double S. So it's COMMISSURES. So just a quick recap. Cerebral hemispheres extending laterally from the diencephalon. At the junction of the developing hemisphere and diencephalon, you will have the roof plate of the diencephalon. Then when you talk about cerebral hemisphere, you have to remember this midline thickening, which is lamina terminalis, which it's a thickening concerned with the commissures developing in the hemispheres. Then you need to understand that this is the cut section of this diagram from this level. This is the third ventricle. This is the roof of the third ventricle, which has choroid plexus. So it's this area here. And growing into the cavity of the third ventricle is developing gray matter of the cerebral hemisphere, which is called corpus striatum. And the corpus striatum is called striatum because it has a striated appearance. We've already done this. And the corpus striatum is important because it divides the nuclei of the basal ganglia. You see the caudate nucleus and the lentiform nucleus. And the basal ganglia are important nuclei concerned with motor activity. So that brings us to an end to the development of the cerebral hemispheres. And now you all know, because you've done head and neck, that when you talk about the cranial fossas and the cranial vault, you talk about different types of meninges, and you talk about falx cerebri and falx cerebelli. So the meninges are three-layered structure. Outermost is the fibrous layer, which is dura mater. Then you have arachnoid and pia mater. All the three meninges, embryologically, they are derived from mesoderm with migration of neural crest cells. Like I said earlier in my lectures, these neural crest cells are very important because they influence the development of almost all the body systems. So neural crest migration is a very important factor, especially in central nervous system and cardiovascular system. Dura mater lines venous sinuses, which you all know that they will drain the CSF back into the venous system through arachnoid granulations. Arachnoid granulations, they project into the venous sinuses, which are lined by dura mater, and CSF is directed back into the venous system. Now you all know that there are 12 cranial nerves. Some are motor, some are sensory, and some are mixed, which means they have motor component, sensory component, and autonomic component. By week 4, all the 12 cranial nerve nuclei are present. Apart from the olfactory nerve, optic nerve, and oculomotor nerve, all the other nerves, they arise in the brainstem. And you must understand that the nuclei or the neurons of these nerves, if they are motor, the nuclei or neurons will be present in the brainstem. If they are sensory, they will eventually come to the brainstem, but through the dorsal root ganglion or a peripheral ganglion. So, week 4, all 12 cranial nerves are present. The olfactory originates in the nose. The optic originates in the eye. It is a continuation of the retinal layer. The oculomotor is present in the midbrain, and all the others arise in brainstem. Now the motor nerves, we are only talking about motor nerves right now. They develop in the area of the hindbrain by thickening or proliferation of neuroepithelium. And this thickening or proliferation is called rhombomeres. You have 8 rhombomeres, and for the 4th, 5th, 6th, 7th, 8th, 9th, 10th, 11th, and 12th. These nerves, they will arise from rhombomeres. Now I'll just show you a picture of the developing rhombomeres. And what are rhombomeres? They are actually thickenings of neuroepithelium in the region of the pharyngeal arches. Can you see this? P1, P2, P3, P4, they haven't showed 6. These are the pharyngeal arches, which we know are mesodermal in origin, and they give off with neural crest cell migration, and they give off a lot of musculoskeletal structures. They have nerves related to each pharyngeal arch. In that region, you will have these rhombomeres, R1, R2, 3, 4, 5, 6, 7, and 8. And you can see that all these nerves, they are developing their nuclei within these rhombomeres. And this segmentation pattern, you have 8 rhombomeres for 4, 5, 6, 7, 8, 9, 10, 11, 12 nerves. These are motor component, the motor component of these nerves. It is going to develop from rhombomeres. The sensory nerves form outside the brainstem from ectodermal placodes. Now we discussed what are ectodermal placodes. They are thickenings for the sensory organs that are developing, like the otic placode, the nasal placode, the lens placode for the eye. And in addition to that, you have epipharyngeal placodes in the area of the pharyngeal arches for 5, 7, 9, and 10. So we'll go back to this. Look at this. In the area of the pharyngeal arches, you'll have 5, 7, 9, and 10. So I'm just going to repeat this again so that you don't get confused. There's nothing to get confused about. Week 4 of development, all cranial nerve nuclei are present. 1, 2, and 3. These are exceptions, the rest all arise in the brainstem. The motor component or the motor nerves, they arise by thickening of neuroepithelium, which is called rhombomeres. These segmentations or rhombomeres are 8 in number, and 4, 5, 6, 7, 8, 9, 10, 11, 12 will arise from rhombomeres. And these are motor components that we're talking about when we talk about rhombomeres. The sensory nerves, those cranial nerves which are sensory, they arise from outside the brainstem or from ectodermal placodes, which can be in the developing eye, in the developing nose, in the developing ear. In addition to that, you have placodes in the area of the developing pharyngeal arches for the nerves of the pharyngeal arches, which are 5, 7, 9, and 10. So cranial nerves, they develop from week 4. The motor component comes from rhombomeres, which are thickenings of neuroepithelium. There are 8 rhombomeres. These rhombomeres are also known as segmentations, and they give rise to the motor cranial nerves and the motor component of mixed nerves. In addition to this, the sensory nerves, they will arise from outside the brainstem, from ectodermal placodes, out of which those placodes which are developing in the area of the pharyngeal arches will be known as epipharyngeal for the 5, 7, 9, and 10th. Now, just a little more and I think we'll be done. Another important structure which is concerned both with endocrinology and gross anatomy when you talk about the abdomen and nervous system is the suprarenal gland. Now, the suprarenal gland has 2 sources of origin. Outside, it has a cortex. The cortex is mesodermal in origin. And inside, it has medulla, which is ectodermal in origin. Now, the diagram that I have put here to make things a little easier for you is that mesothelial cells, or actually cells which are going to transform into mesoderm. These mesothelial cells, they will start to increase in number, they will proliferate in the area which is going to be between the mesentery, which is this part here, suspension of the gut, and the developing gonad, which is this urogenital ridge. So between the root of the mesentery and the gonadal ridge, you will have proliferation of cells, and these proliferation of cells will aggregate and they will transform or evolve into acidophilic cells, and they will form the primitive cortex. So now we're talking about the suprarenal gland. It has a cortex which is mesodermal. It has a medulla which is ectodermal. Actually, the medulla is neuroectodermal. Week 5, cells will proliferate and you will have the developing suprarenal gland. And the initial cells will form a primitive cortex which will have acidophil cells. This is the diagram that I want to show you. This is the acidophilic cells, and they will form the fetal or the primitive cortex. The second wave of cells in the area of the cortex will be smaller in size, and they will form outside the primitive cortex, and then this will be known as definitive cortex. This area, this is fetal cortex. Afterwards, you'll have a second wave of cells, and they will come to lie outside here. and slowly the initial acidophilic cells will go away or they'll transform, and they will then form the definitive cortex or the adult cortex. And the adult cortex has three zones which you study in detail in histology. Now, to remember these zones from without inwards, glomerular filtration rate, GFR, zona glomerulosa, zona fasciculata, and zona reticularis. We are talking about the cortex of the suprarenal gland. Initially, you have in the cortex acidophilic cells which will form the primitive cortex. Then a second wave of cells, they'll form the definitive cortex. These cells will be smaller in size than the original acidophilic cells. They'll be steroid producing and they'll produce dehydroepiandrosterone, which is DHEA, for production of estrogen and androgen. And the definitive cortex from without inward will have three zones: zona glomerulosa, zona fasciculata, and zona reticularis that you see here. Now we come to the medulla. The medulla cells from the neural crest will be migrating and invading this developing suprarenal gland medially, from the medial aspect. And these neural crest cells will then form the medulla. And how will they do it? These neural crest cells will come into the medial aspect of the developing suprarenal gland and they'll form the medulla. These neural crest cells will transform into chromaffin cells, which are a modification of the sympathetic cell neurons. And these chromaffin cells of the medulla, they'll be innervated by postganglionic sympathetic fibers. When these fibers are activated, they will produce epinephrine and norepinephrine. So these chromaffin cells will form the medulla, and these chromaffin cells will secrete epinephrine and norepinephrine. I think that now it's been almost 40 minutes since we started the lecture. So the next thing that I want to explain to you, I think we'll do it in the next lecture. And I hope you understood what we did so far. And the rest of the lecture and the rest of the topic that is left, we will discuss in the next lecture. Thank you and Khuda Hafiz. अभी मुझे एक दफ़ा सुना